All right, so glycogen is the animal energy storage molecule. This is the fuel storage. This is all alpha glucose. Structurally, it is very similar to amylopectin, but it is more densely packed. There is more branches of the alpha 1,6 linkage that we discussed before. That alpha 1,6 linkage, I am going to simplify that drawing. All right? That will be our 1,6 linkage. That is our alpha 1,6 linkage. All right? That's our alpha 1,6 linkage. And the whole thing will continue from there. So we have more branches, more closely packed, and we have shorter chains by right of them being more closely packed. So if you have more branches at greater frequency, you will have shorter chains of alpha 1,4 glycosidic bonds. So let's talk about the structure of cellulose now. We discussed earlier with our beta glycosidic bonds, with our beta 1,4 glycosidic bonds, where we have the up, the down, etc. So because of this, we will have a hydrogen bond here. Oops. CH2OH, CH2OH, versus a 1 CH2OH. So here we have this hydrogen bond there, and we also have this will be our all right. See, we have the OH, 2 hydrogen bond there. Hydrogen bonds, remember, are always described as are always written and drawn as a dashed line. All right. So this alternating pattern of up, down, up, down, up, down, up, down, up, down, allows for these chains of beta glucose to hydrogen bond to one another. The tightness of these hydrogen bonds effectively, well, it's a branded product, but whatever, Ziploc sealing to one another, is going to prevent water from being able to leak through a fiber. A cellulose microfibril will be water insoluble. Beta glucose cannot dissolve in water. Water cannot pass through these tight junctions between these hydrogen bonded fibers of cellulose. But the individual microfibrils are going to be arrayed in such a way that they will effectively be full of holes. So cell walls, which are primarily composed of layered microfibrils of cellulose, are going to be full of holes. They're going to be porous. So they will be freely permeable to water, and water can adhere, water hydrogen bonding to a substance that is not water, to cellulose microfibrils, in order, and this allows for the apoplastic pathway when we discussed Unit 4. It also allows for the transpiration, adhesion, and cohesion forces later on, also in Unit 4. All of this stuff will be seen again. Now, typically, a carbohydrate monosaccharide are those with ratios of C1H2O1. This is what I told you. But you can dope in the sense of metallurgy, where you add a carbon and iron to make steel, for example, carbohydrates with other elements to change its properties. For example, adding nitrogen to a carbohydrate will give you N-acetylglucosamine. N-acetylglucosamine, which has a chemical formula of C8H13O5N, that repeats N times. You can have this unit repeat multiple times. This polymer gives you chitin. Chitin is the shell material in crabs, in beetle arthropods, in any insect or arachnid or whatever that you find. This is going to be N-acetylglucosamine. So that's N-acetylglucosamine. That is a carbohydrate. Glucosamine is a carbohydrate, but it has nitrogen in it. That can be a question. Do not be fooled. Now let's talk about lipids. Amphipathic lipids are those lipids that have a polar head and a non-polar tail. Fatty acids will have the formula CnH2nO2. These are carboxylic acids with a hydrocarbon tail of variable length. Attaching a compound to the carboxylic acid end will give you soaps and detergents. So if I add length to this tail, so if I make this a hydroxyl group here, and I make this a carbon tail length that just continues on and on and on like that, that's fine. If I shorten the tail like that, that's also fine. Whatever, it's still a fatty acid. There are two types of fatty acids in this world. There are saturated and unsaturated. All it takes to be unsaturated is to have a single double bond in your hydrocarbon chain. By single double bond, I mean you only need to have one, how to phrase this without using the word single. You only need to have a double bond found somewhere within the hydrocarbon chain. That's a better phrasing. Let's use that. Saturated. A saturated fatty acid is one where there are no double bonds. Every carbon is bonded to at least two hydrogens. At least two hydrogens are bonded to every single carbon. If you have a double bond, these two carbons here that are participating in that double bond are only bonded to one hydrogen. It is not possible for them to bond to anything else. So that is a saturated and an unsaturated. Unsaturated. They could be saturated if you had more hydrogen. A saturated towel is one that cannot hold any more water or fluid or whatever. So a saturated hydrocarbon chain is one that cannot hold any more hydrogen, no matter what. Now, your saturated fatty acids are going to be your butters, your eggs, your meats, your fishes. Your unsaturated fatty acids are going to be your oils, your fruits, your vegetable oils, all those things, your peanut butters, which is also vegetable oil, by the way. So, which is greater? The melting point of a saturated fatty acid is higher than an unsaturated fatty acid. This has to do with a property in organic chemistry of similar to like dissolves like, where polar compounds will fit better together if they are unsaturated. But as soon as you put a double bond in that chain somewhere, eventually what you're going to end up having is you're going to have more space right here, and they're going to separate more, and they're going to have less of a van der Waals force interaction, and less interaction between molecules means that they will separate more easily at lower temperatures, which means they will have a lower melting point. So an unsaturated fatty acid will have a lower melting point. The density of a saturated fatty acid you can fit more of these fatty acids together if they are saturated than you can unsaturated fatty acids. These will take up more space because they are bent and kinked. Reactivity. Saturated fatty acids are less reactive than unsaturated fatty acids because double bonds between carbons are more reactive from a chemical standpoint. But therefore when you burn them, when you use a chemical reaction combustion to burn saturated fatty acids like fat, whale blubber or whatever, kerosene for example, it burns very well. A very high energy, a very high temperature flame. So you will have a higher reactivity, got that backwards. It'll be a higher temperature flame with oil than it will be with whale blubber. You'll have a lower intensity, longer burning flame with whale blubber than you will with a oil. So a grease fire will be more energetic than will be a penguin blubber fire. And by the way, yes, you can actually have a fire with nothing but penguin meat. It is that fatty and oily. It is terrible. It's supposed to be very bad tasting. I've never had it myself. But conversely, when you metabolize it, when you yourself eat these substances, you get more energy. You get more ATP by using saturated fatty acids as fuel for aerobic metabolism, aerobic respiration, than you do for unsaturated fatty acids. And the reason for this is that your body, your living things, your living cells, they are going to utilize energy from carbon-carbon single bonds. The proteins and enzymes that are in your cells are set up for the breakdown of carbon-carbon single bonds. So you get more energy more efficiently from saturated fatty acids than from unsaturated fatty acids. So calorie for calorie, unsaturated fatty acids are better for you. That's why they keep saying that plant fats are better for you than animal fats. That's why you should eat more plants than animals. Now let's talk about non-polar lipids. Those were amphipathic lipids that we just talked about. Non-polar lipids are things like waxes. These are esters of fatty acids. If you remember, an ester is a carbon-oxygen-carbon bond. So, carbon, oxygen to carbon. So you have your carbon chain over here, your carbon chain over here. A wax will have that going on over there, but steroids can be recognized by their tetracyclic skeleton, which means 4-ring skeleton. They are consisting of 3 fused 6-membered rings and 1 5-membered ring. So one, oh wow, that looks terrible. Let's fix that. Nope, that is also terrible. To make that look good. All right. All right. There we go. Other stuff will be attached to it, but that other stuff varies and is not something you can be tested on directly. So, this is our lax blah blah blah. Now, we also have triglycerides. Triglycerides are a non-polar fatty acid. Effectively, you take this and you're going to get this as your simplified diagram. This simplified diagram, you have your glycerol. Glycerol is carbon, hydrogen, hydrogen, carbon, hydrogen, carbon, hydrogen, hydrogen, hydroxyl, hydroxyl, hydroxyl. Bonded to three of those. Does this look familiar to you? I certainly hope it does. Condensation reaction will form this. It will be formed by a condensation reaction. Water will leave and you will be left with a carbon-oxygen-carbon double bond, an ester, forming a fatty acid. A glycerol with 3 fatty acid tails. This will happen 3 times, you'll get 3 waters. Now, triglycerides. These are the storage forms of lipids in animals and plants in the forms of fats and oils. The food items that were shown as examples of saturated and unsaturated fatty acids were actually examples of fatty acids clumped into triglyceride units. So, in animals, your triglycerides will typically be saturated triglycerides. And your plant oils will be unsaturated triglycerides. They will consist of unsaturated fatty acid tails on a triglyceride, glycerol with 3 fatty acid tails. They are a fantastic source of energy. They are a fantastic source of fuel because they have many carbon-carbon single bonds. They are highly reduced, so you can release this energy by oxidation. They can be localized in an organism due to their insolubility in water. Because they are not polar, they will not dissolve in water. They will not easily diffuse throughout the cell. They will stay wherever you put them. So, if you have a cell and you want to store triglycerides, if you have your cell over here with your nucleus over here, and you want to store triglycerides, you can make a clump over here, unbound by a membrane, and just have a clump of triglycerides. Not Cambridge appropriate, because I labeled inside the diagram and I shaded. Unacceptable. So, this doesn't even need to be bound by a membrane inside the cell. They will clump together because nonpolar will dissolve nonpolar. It will clump together, like dissolves like. And the polar water molecules around it will form this bubble. So you can just sequester triglycerides within a cell. You don't need to do anything special to store them. So, it's a condensation reaction. Well, I've already said this. It's 3 condensation reactions, actually. Now, how do we turn a triglyceride into an amphipathic lipid again? We add a polar group. So, we have 2 fatty acid tails, and we have a phosphocholine group. This is the beginnings of our phospholipid. Our phospholipid is extraordinarily important when we'll start discussing in Unit 2 the plasma membrane, which is primarily composed of phospholipids. So here, we'll have our glycerol as our backbone. We will have 2 fatty acid tails, whether they are saturated, as shown here, or unsaturated with kinks in the chain here and there. Doesn't matter right now. We're not working with that. And one of those spaces will have a phosphate group, which is negatively charged, and a choline group, which is positively charged. Having a positive and negative charge in one clump, this is a polar molecule, which has a positive and negative end. That makes this a polar molecule over here, and a non-polar set of molecules over here, but they're attached as one molecule, so you have a polar head and a non-polar fatty acid tail. Polar phospholipid, polar phosphocholine head, non-polar fatty acid tails. Cholesterol. We can add to a steroid compound, that tetracyclic ring, a simple hydroxyl group right here, and suddenly this whole molecule is polar. This one group, which has an electronegative charge, which has electronegative difference in charge, from a ability to form hydrogen bonds, makes this a polar head and this a non-polar tail. So what does that really get us? It allows us the ability for water to hydrogen bond with the polar groups of cholesterol and the phospholipid heads on either side of a phospholipid bilayer, and for the fatty acid tails to interact with the cholesterol of the nonpolar cholesterol tetracyclic ring here. So we have nonpolar barrier to polar solvents on either side. This is where we go with that. And now we can move on to amino acids. So here is the structure of an individual amino acid. I showed this to you earlier before. We have an amino acid group here. We have our alpha carbon in the center. We have our variable side chain R here. And we have our carboxylic acid group here. Protein structure begins with peptide bonds. Peptide bonds are always formed between the amino group and the carboxylic group. They will form this way and only this way, because the amino group will gain a hydrogen and therefore gain a positive charge, and the carboxylic group will lose a hydrogen and therefore lose its positive charge, gaining a negative charge. So they will line up along their positive and negative poles. Positive to negative, positive, negative, positive to 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 negative. They will line up in this way. Then we will have a condensation reaction occur, where we have, I'm going to shorthand this, CO minus H, and